Oh, great.
Cupcake. And,
This guy?
Let's move on.
Got a blue one.
So we have now two more talks this morning. One, is it two or three? Two. One by Matt Spitzer
three.
now, and then we're also going to hear about Cytobank.
Yeah, we'll do that for a while.
Okay. And that's it. Matt, you're up.
Morning, everyone. My name is Matt Spitzer. I'm a grad student in Gary's lab, and as he said, I've been there for just a few months, so if there's anything to take away from this talk, it's really how quickly you can get up and running using this mass cytometry platform. And also to note a lot of the concepts that I'll be discussing here in terms of reagent development, and especially the titration segment, are also very applicable to work that you would do on a normal fluorescence platform. So for those of you who don't anticipate doing mass cytometry in the near future, don't tune out. So the topic, obviously, as you see, will be focused on reagent development with an emphasis on the antibody conjugation process that we do in the lab to get reagents up and running for mass cytometry. A brief overview of where this talk is headed. I'll start with a reasonably in-depth discussion of the antibody conjugation process, including an overview of some of the chemical principles that are employed, a reasonably in-depth discussion of the protocol itself, some helpful tips when you're doing your titrations, things to think about, things to consider, and then some tips for troubleshooting when things inevitably go wrong every now and then. And then I'll touch briefly at the end of the talk on some other tools that are in use in our lab and elsewhere, some viability stains and some additional reagents as well. So taking a note out of Sean's book, a brief refresher, how do we get up to 50 parameters or above in theory? Well, we take advantage of the fact that heavy metals, the non-biologic elements on the periodic table, are really heterogeneous compositions of multiple isotopes. Gave you the example of europium having two stable isotopes, 151 and 153. And if we look at a full nuclide table, this really applies to the vast majority of elements here in the heavy metal non-biologics, such that each of those vertical bars, different stable isotopes, are ones that can be measured as individual parameters, individual channels in a mass cytometry experiment. So where do we actually stand? What's in use day to day in our lab and elsewhere? Well, antibodies, you'll see, are largely reliant on the lanthanide series, which, as you all know from your introductory chemistry, is part of that little table at the bottom. Always wondered what it was good for. Well, here's what it's good for. And an additional indium. And these are really the elements that we'll be focusing on in terms of antibody conjugation. And that's for a few reasons, but largely because they all form trivalent salts, which are easily conjugated through the chemistry that we use and that's been developed by the folks at DVS, which is the company, as you know, that has developed the mass cytometry platform. So if you'll take a look at the bottom panel here, a brief orientation as to where we're headed and what the goal is for antibody conjugation. We start with a purified antibody, which will determine our bind to our marker of interest. And to that, we'll be attaching a polymer chain, which includes in it a series of chelating groups which bind with a high affinity and coordinate in their center a trivalent ion of one of those lanthanide metals. So the chelating group itself is DTPA. It's diethylene triamine pentacetic acid. For chemists in the room, you can see the structure there. And in the center, it coordinates your metal ion, the trivalent lanthanide. And it's important to note that the dissociation kinetics here are actually an order of magnitude lower than what you'd see for an avidin-biotin interaction, which in a biological context we think of as being essentially irreversible. So this reaction really is going to completion, which is fantastic. And using these polymers that are commercially available from DVS, we currently attach between 1 and 300 metal atoms to each antibody. And those ions then are what are passing through your ICP-MS and giving you the signal readout. So how do we couple the polymer to the antibody? Brief introduction to this chemistry. It relies on a bis-muoyamide linker, which you see there, the polycyclics. And in order to link that up, it relies on a partial reduction of a sulfide on the FC portion of the antibody to which it attaches. And then this links into the polymer chain with the chelating groups that I've just described. And this reaction also approaches completion when we use the polymer in excess. So through these steps, which are both give you reasonably high yields, we're able to recover the majority of the antibody that we begin with, which is great considering how pricey they can be. So where do we start? How do we choose a good clone to conjugate for mass cytometry? Where we like to start, in the Nolan lab at least, is to use antibodies, if possible, that have already been tested and validated for use on a fluorescence platform for traditional flow cytometry. And we do this for a couple reasons. The first being that it provides a great means of comparison. You can toss the same clone onto cells and stain them for fluorescence and for mass cytometry simultaneously, run them, and ensure that the conjugation process isn't actually destroying the specificity of your antibody binding. And secondly, that these clones are generally robust. They have to be conjugated to fluorophores, and they have binding characteristics that are favorable for these sorts of experiments with high specificity as well as reasonably high affinity. That said, it is possible to develop antibodies against targets for which there are not commercially available flow cytometry antibodies. And as you'll see through the talks later this afternoon and into tomorrow, it's been done before. It's more challenging, as you might imagine, and you do have to start with your purified monoclonal against whatever target you're interested in. And the validation really requires that you have an established positive and negative control population to determine that what you're measuring is actually what you intended to measure, which is always a good thing. And that can be more difficult for some markers than others, as you might imagine. So now we'll dive into the conjugation process itself. You're sitting down, what do you need to start with? Well, obviously your antibody. And it's great to have this in a carrier-free form, purified. You don't want any BSA in that solution in order to ensure that the efficiency of your reactions is really maximal. What we generally prefer to use are IgGs. This reaction tends to work better than for IgMs. It is possible to label some IgMs, but there's heterogeneity there. So IgGs if possible. And the protocol that we use has been optimized to label 100 micrograms of antibody at a time, which at the end of the day will give you enough reagent for several hundred tests when it comes time to do your experiments. So a stock, depending on how active you are in the lab, can last you quite a while. You'll also need the polymer, which is commercially available from DVS under the trademark MaxPar. And we use 200 micrograms of that per labeling reaction. And then the lanthanide itself, which comes in the form of the trivalent salt. So that's the lanthanide trichloride. And we keep that in our lab in stock solutions between 50 and 100 micromolar in L-buffer, which is the first of a 4-part series of buffers that come as a kit from DVS. They're loading buffer, reducing buffer, coupling buffer, and washing buffer. And it's important to have all of these. They're important for different parts of the antibody conjugation process to ensure the efficiency as you go throughout. So that kit you can purchase. And then the reducing agent, as I mentioned, the antibody conjugation relies on reducing a disulfide bond in the Fc region of the antibody. And that reagent is TSEP tris-carboxyethylphosphine, which is a relatively mild reducing agent, important to avoid breaking the disulfide bonds that are important in the FAB regions of the antibody for maintaining antigen specificity. And lastly, to store the antibody, keep it in good working order in the fridge. You'll want antibody stabilization solution, which is available commercially in either TBS or PBS formats. And then a few materials that you'll want at your beck and call before you get started. The first being millipore centricon columns. These are filter columns, and you'll want 2 different sizes, a 3 kilodalton and a 50 kilodalton filter. Great for bringing up materials in different buffers and washing and those sorts of things. Some screw top epis, and then filter tips. And it is important to use filter tips whenever you're doing anything involving mass cytometry to avoid cross-contamination of your metals. As you can imagine, labeling several antibodies at once, you get a little bit of metal solution up on the bottom of your pipetter. stick it into a different antibody, and all of a sudden what you think you're measuring on one channel is spread across 3 channels, and you've really lost all sense of specificity. A few notes and considerations to think about as we walk through this protocol at sort of a high level. The first, concentrations are very important throughout. The antibodies can actually precipitate if they're exposed to a high concentration of those trivalent metal ions, as you might imagine. They can strip the antibodies water and pull them out of solution. The fact that the antibody is purified is important. So if a purified antibody is not commercially available for your target, you should go ahead and go through the purification steps yourself to ensure that any carrier proteins in that solution, most commonly BSA, are not present when you do try to label the antibody. And then the timing, since we're partially reducing the antibody and also chelating those metal ions, the timing throughout is reasonably important. So if you do decide that mass cytometry is in your future, there is a detailed protocol that is important to follow. And now we'll walk through that protocol. Yeah?
Sodium azide a problem?
No.
No.
During the conjugation itself?
Just, most antibodies come with sodium azide, so when you're doing typically labeling, which is amino-based, it can be an issue, but it doesn't appear to be a problem here.
It's not a problem during the labeling process, and we actually store our antibodies in azide to prevent microbial growth and that sort of thing at the end of the day also. So we'll take a brief overview of the protocol itself, just to give you an idea of the steps that are involved. This is something that from start to finish can really be achieved in 4 or 5 hours, and the titration takes a few hours also. So 1 full day, and you can have new tools in your toolbox, which is fantastic. So the first step will be to chelate those metal ions onto the polymer, followed by the partial reduction of your antibody separately. And then adding these 2 reagents together will form our fully conjugated antibody that's ready to use on the mass cytometry platform. So the first step, preloading our chelating polymer. This is done in the L buffer, the loading buffer, that is available from DVS. The first step is to go ahead and bring the polymer up in solution, add your trivalent lanthanide stock. We keep ours at 50 micromolar, and we add about 5 microliters of that stock. And incubate that for an hour at room temperature. Again, as I mentioned, this reaction essentially goes to completion, and what we have at the end of the day is a polymer that is chelating the metal ions that will give us our signal when it's passed through the ICP-MS. Separately, but simultaneously, we will be taking our antibody and bringing it up into the R buffer, the reducing buffer, and adding a relatively dilute 4 micromolar concentration of TSEP, which is the mild reducing agent that I mentioned previously. And the goal of this step is to partially reduce the antibody to expose that sulfide in the FC portion for derivatization. So let's take a look at what exactly is going on here. If you look at the structure of an IgG, you'll see those red bars are the disulfide bonds that hold this guy together. And there are several in the FAB regions and in the FC regions. Importantly, the disulfides in the FC regions are actually more easily reduced than those in the FABs, which is the principle that allows us to conjugate these antibodies in the first place using that bismulamide linker. And those disulfide bonds are also holding the 2 arms of the antibody together in the linker region. So what may be happening is that those antibodies can partially break in solution, but since the protocol is optimized to leave intact the disulfides in the FAB region, the binding specificity will be maintained throughout. So it's not worrisome as long as the steps are followed carefully. So that reduction takes place over 30 minutes at 37 degrees. And then the last step in the coupling buffer will be to combine, after several washes, both the partially reduced antibody and the chelating polymer. And after 2 hours at 37 degrees, you'll have what is hopefully a functional antibody conjugated for use on the mass cytometry platform. So relatively simple. I think the chemistry is not too difficult to grasp. And recapping a bit, first step, chelate the metal. Second step, reduce the antibody. Third step, combine these two together to produce the conjugated mass cytometry antibody. So now that we have an antibody, what's next? Well, we generally like to quantify it to determine what concentration we have and what our yield was. So we do this using a spectrophotometer like a nanodrop, measure the absorbance of IgG at 280 nanometers. And what we expect at the end of this process is generally a yield in the range of 60%. So for the 100 micrograms of antibody that you start with, you'll end up with about 60 at the end of the day, which isn't too bad. Upon quantification, we generally dilute our antibody in that stabilization buffer to a working stock of between 0.1 and 0.4 micrograms per mil. And we use that to do our titration then, which I'll be walking you through in some detail. The goal of the titration is to determine the optimal dose for staining for each of your individual antibodies, which, as Peter mentioned in his talk, is very important when you're using permeabilized cells to ensure that you're really looking at what you think you're looking at. So a lot of this applies, as you might imagine, very similarly to titrations for phosphoflow on the flow cytometry fluorescence platform. And we use a 7-step titration scheme ranging from 8 to 1/8 micrograms per mil for our mass cytometry antibodies, in a similar range to what you would use for a fluorescence conjugated antibody and stain in a residual volume of 100 microliters. And importantly for our titrations, we like to have predetermined positive and negative controls so that we can ensure that we are getting specific staining. So here, I'll show you a few examples of antibodies that I've labeled and walk you through some of the considerations to think about when you're titrating your antibodies. I'm working primarily in mouse, most conducive to the sorts of questions I'm interested in asking. And if any of you have read Sean and Aaron's science paper, you'll know that a lot of the work that's been done previously on mass cytometry is in humans. So these are all new reagents that I'm working on. And what I've done here in mouse experiments, for those of you who work in mouse, you know that congenic markers can be very valuable for adoptive transfer experiments. You stick cells into an animal and you want to know whether they came from the donor or from the recipient. And CD45 is one of those congenic markers that we all know and love. So I've labeled CD45.2 here. I had previously labeled CD45.1. And my goal here is to distinguish between these two populations to be able to determine what's happening with my lymphocytes after injecting them into a host. So as you'll see here, I've labeled cells with both of those. And looking just at a 50-50 mixture of those cells from in vitro, you'll see that as I label CD45.2, I have a nice bimodal distribution of those cells in my singlet gate. So I have both my 45.1 and 45.2 positive cells. And you'll see as I increase the dose of antibody, I have a titration curve that you might expect. The staining increases. If you look at the 45.1 positive cells, you'll see that they're all essentially 0 for CD45.2, going back to Sean's point that essentially there is no background on the mass cytometry platform for the majority, I would say, of different epitopes that we'll be looking at. And then looking at my 45.1 cells, you can see, if you look at the values on the table on the right, that there is a diminishing return, as you might expect. The beginning, the lower level of that curve, as you increase the concentration of antibody twofold, you actually do get about a twofold bump in your signal. And that steadily decreases over time. So if my intention here were to merely separate my 45.1 and my 45.2 cells, you can see that a concentration of about 1 microgram per mil is probably sufficient to do that. And I'm not losing too much signal, even if I were to increase that 8-fold. Something else to consider more antibody isn't really always better, especially when you're working on fixed permed cells, as Peter mentioned. So here I've labeled anti-CD3, and I'm showing you biaxials with CD3 against B220. So CD3 obviously being our T-cell marker, part of the TCR signaling complex, B220 being a B-cell marker in mouse. And then at the bottom there, CD3 by CD90, both of which are T-cell markers. So I'm looking at my double positive population there. And what you'll see is as I titrate up my concentration of antibody, really not getting a tremendous differential between the positives on my T cells. But what is happening, if you'll take a look at those boxes on the upper portion of the charts, is that my background staining on my B cells is actually increasing pretty substantially as I add more anti-CD3 antibody. So it is really important to monitor the upper end of that titration curve as well to ensure that what you're measuring is really a specific epitope that you're intending to measure. Again, stressing that that full titration range, the 7-step that we use, is really ideal. And another thing to consider, here I've labeled CD44, which is a memory marker in T cells. So you see the bimodal distribution between my naive and my memory T cells up in the upper concentrations. But you'll also see that as I increase the titer of antibody here, that I do see that diminishing return effect, such that increasing from 1 microgram up to 8 really doesn't give me much of a difference in the staining on my memory cells. But depending on how important looking at memory cells might be in your experiment, or whatever marker it is you're looking at, you could imagine that your specifications could differ pretty vastly. So again, it's important to really sit down with your own experiment, think about what you're trying to measure and what your hypotheses are, what's most important to you, and optimize those reagents for yourself rather than taking anyone else's word as gospel. A few tips on troubleshooting when things go wrong. The first thing that I like to do is to go ahead and stain cells with my CyTOF conjugated antibody and come in with a fluorescence conjugated secondary and run that on a fluorescence platform on a fax. And what that allows me to do is to determine whether I maintain specific staining through the conjugation protocol or not. If I see that my antibody is still labeling the antigen that I was hoping it would, then that's generally good news. It just means that something went wrong in the conjugation process, and I'll repeat it. If, on the other hand, I don't see the specific staining that I was hoping for on a fluorescence platform, what often happens is that through that conjugation process and the reduction, the specificity of my antibody has been destroyed, which is always unfortunate, but we do have a few tools and methods that we can use to circumvent that problem. The first being to use a second clone, obviously, if the antigen you're trying to measure has different monoclonals against it, just try a different one, see if it works. If not, there are a few systems that we and the folks in Mark Davis's lab have optimized to get around that problem. The first being a biotin streptavidin system, where we use a biotinylated antibody and a metal conjugated streptavidin secondary to circumvent the need to conjugate the monoclonal itself. And the second strategy, we can use an anti-fluorophore secondary, where we'll use a fluorophore conjugated primary antibody, something like PE or APC generally, that is a large protein fluorophore, and then come in with a anti-fluorophore secondary that's conjugated for the CyTOF, another approach to circumvent the need to actually label your primary monoclonal itself. So now I'll give you a brief introduction to where we're headed, or where we are hoping to head in terms of these reagents. As I mentioned, this chemistry that we currently use and that folks are using really around the world now can label up to 100 to 300 metal atoms per antibody, which is sufficient for a good portion of the needs in terms of specificity and sensitivity that we have, but isn't really as ideal as some of the most quantum efficient fluorophores on a flow cytometry platform. In order to increase the number, what folks are thinking about or working on is a nanoparticle-based approach. So the Q dots, which were introduced in flow cytometry some years ago, are nanoparticles you might know and composed of a cadmium core. And that cadmium is not isotopically enriched on a Q dot, and it ranges in mass from 110 to 114, which does fall in the mass window of the CyTOF. So we can actually measure Q dots on the CyTOF. And as you'll see there, the Q dots that produce wavelengths of light in the higher range, the red Q dot at Q dot 800 and 655, the orange, are actually composed of a larger core of cadmium. And that is proportionally increased as signal when you look at it on the CyTOF, since those metal atoms are what we're actually measuring. And this strategy was used by Sean and Aaron in their science paper to measure CD3. As you'll note, the lack of isotopic enrichment required them to actually sum the signal over the 5 different cadmium channels, 110 to 114. And additionally, because these are in the low range of the mass of the sensitivity spectrum of the machine, they chose an antigen that's highly expressed, CD3, and were able to successfully gate their T cells. So potentially nanoparticle labeling may be in the pipeline. And with that, we'll leave antibody conjugation. And if you have any questions about specific markers that you might be interested in labeling, I'd be happy to talk to you about them. And we'll discuss a few other tools that we use in the lab to essentially bring cytometry up to an even playing field with flow cytometry. The first being our viability stain. So you may be familiar, for those of you who do flow cytometry, with the AquaLive dead stain that's very widely used to gate out dead cells before performing an analysis. And what we use for mass cytometry is cisplatin, which you may be familiar with as a chemotherapeutic agent. It's actively transported across the plasma membrane of live cells, but dead cells with permeabilized membranes can take up cisplatin fairly quickly. And as you'll see in the bottom panels, the level of staining that we get on AquaLive dead on a flow cytometry platform or using our cisplatin viability stain on a mass cytometry platform are actually very comparable. So if investigating live cells is very important for your experiment, then that's another tool in the toolbox that you can utilize. A few additional reagents Sean mentioned are DNA intercalators that are commercially available from DVS, either the iridium or the rhodium-based intercalators, and those are very useful for measuring cellular events as they pass through. And lastly, we have a few barcoding reagents that we use to stain multiple samples across conditions or across individuals simultaneously, run them through the machine at one time, and then afterwards deconvolute that data back out and determine which conditions the cells were originated from. And you'll hear a much more lengthy description of those reagents from Eli Zunder tomorrow. And with that, I'll point out the folks in the Nolan lab, as well as the team up at U Toronto and at DVS, led by Scott Tanner, who have really developed all of the reagents that I talked about. And I'd be happy to take any questions if there are. Yeah?
Is there a way to measure the range and distribution of the number of molecules when you conjugate the antibody? Since theoretically then you could actually work back to an absolute number of phosphorylation events or like binding.
You mean the number of metal atoms per antibody, or the?
Yeah, because then you could take your intensity on the cytometer and potentially work back to the absolute numbers within the cell, correct?
Well, in theory, I mean, you're sort of coming into the same problem that you have with flow cytometry in terms of the saturation of that process and knowing exactly what proportion of molecules you're staining. I mean, the short answer is no. There is no easy way to determine how many metal atoms are on any given antibody. But even if there were, you'd sort of hit the same roadblock that you would with FACS in terms of doing that.
I just want to add one thing. So you can use a plate-based ICP-MS to get an average of that number, but of course, it's just like flow-to-protein ratio via distribution.
Yeah.
And then, as Matt said, you don't know if you've really stained every epitope on the surface of that cell, so it also depends on the performance of your antibody. But it's a direction that we're trying to go. I mean, it's kind of the dream.
Yeah.
One of the great things about this technology is that it's very quantitative, so
Yeah, that's what I was thinking.
potentially to go that
That is true.
Yeah, it's just not there yet.
All right, then I'll turn it over to Nikesh for, yeah, one more.
Everyone else does.
Yeah, go for it.
Is there any thought of developing a labeling kit for oligos? Because then you could potentially also probe some transcripts at the same time.
It is something that I'm very interested in and currently working on. So we can chat. Yeah.